Good afternoon, everyone. Today we're going to explore CXCR4-mediated metastasis in Foxi3 ablated prostate cancer cells. So pretty much how Foxi3 and CXCR4 may work together to influence whether prostate cancer becomes metastatic. We'll walk through what cancer is, how it spreads, what EMT is, and why Foxi3 and CXCR4 might be connected in ways that shape cancer behavior. Let's start with the big picture. Prostate cancer is extremely common, and 1 in 4 men will die from it. Most cancers begin quietly. They grow slowly inside the prostate gland, and may cause symptoms for years. But the real danger begins when cancer cells learn how to break away from the prostate and travel through the body and establish new tumors, especially in the bone. So understanding how cancer spreads is essential for developing better treatments. To understand metastasis, we need to know or understand what cancer actually is. Cancer is not just uncontrolled growth. It's a disease where a normal cell accumulates changes over time. These changes allow the cell to break the rules that normally keep the tissues healthy. On the slide, you can see the progression of normal cells to abnormal cells multiplying, to malignant cancer, and then angiogenesis, and lastly, invasion. Cancer cells gain several dangerous abilities. They can divide uncontrollably, resist cell death, ignore growth suppressors, become essentially immortal, stimulate angiogenesis, meaning they force the body to grow new blood vessels to feed them, and eventually they gain the ability to invade and metastasize. These abilities don't appear all at once. They accumulate as cancer hijacks more and more biological systems. Cancer hijacks systems that normally protect us. It hijacks growth signaling so that it can divide without permission, growth suppressors so it ignores the brakes, apoptosis pathways so it refuses to die, angiogenesis so it can build its own blood supply, and lastly, cell identity programs, so cellular differentiation so that it can change its shape and behavior. This last hijacking changes the identity, which leads us to EMT. So what is EMT? One of the most important processes that cancer hijacks is epithelial mesenchymal transition, or EMT. Under normal conditions, EMT is a healthy process. It helps with wound healing, embryonic development, and tissue remodeling. But in cancer, EMT becomes a weapon. Epithelial cells are normally tightly packed, strongly adhesive, stationary, organized into layers, and play a role in forming barriers. Mesenchymal cells, on the other hand, are loosely organized, weakly adhesive, spindle-shaped, highly motile and invasive, and play a role in tissue remodeling, wound healing, and migration. Metastasis is the process in which cancer cells break away from the original tumor and they travel throughout the body, learning how to form new tumors in distant organs. This is not a random process. Cancer cells follow chemical signals, almost like a GPS system. One of the key molecules involved in this guided system is the chemokine CXCR4, which is the chemokine receptor CXCR4. CXCR4 normally helps cells move to where they are needed. But in cancer, CXCR4 becomes overactive, and high CXCR4 levels help cancer cells. So they help these cancer cells to invade surrounding tissue, stimulate angiogenesis, protect themselves from apoptosis, and migrate toward organs rich in its ligand, which its ligand is CXCL12. In prostate cancer specifically, CXCR4 is strongly linked to metastasis, especially to the bone again. Prostate cancer is one of the most common cancer types in men aside from skin cancer. It usually grows very slowly at first, but becomes aggressive very quickly. Bone is the preferred metastatic site for prostate cancer, as more than 90% of patients with metastatic disease have bone involvement. This makes understanding the molecular drivers of metastasis extremely important. Now let's introduce the second major player, Foxi3. Foxi3 is a part of the 4-KIT box family of transcription factors, in other words, Fox family. These proteins regulate cell differentiation, meaning changing of cell types, cell proliferation, uncontrollably growing or uncontrollable growth, tissue development. Lastly, Foxi3 has been associated with regulation of genes involved in EMT, which means that it may influence how cancer cells transition into more invasive states. Foxi3 expression has been observed across multiple cancer types. This suggests that Foxi3 may play a role in regulating pathways that cancer cells hijack, not just in the prostate or not just in prostate cancer, but across a spectrum of malignancies, such as breast, head and neck, liver, lung, melanoma, ovarian, prostate, thyroid, etc. 3 is also important in bone biology. Studies show that in mice and dogs, Foxi3 deficiency leads to abnormal bone and dental development. This tells us that Foxi3 is also deeply involved in bone morphogenesis, meaning how bones acquire their shape, structure, and organization. In humans, Foxi3 mutations cause craniofacial abnormalities and congenital oral atresia. Again, this reinforces that Foxi3 regulates developmental programs, including bone and cartilage formation. This is interesting because prostate cancer metastasizes to the bone, so a transcription factor involved in bone development may also influence how cancer interacts with bone. Both Foxi3 and CXCR4 have been implicated in prostate cancer progression, so this raises an important question for me. Could Foxi3 regulate CXCR4? If so, Foxi3 might influence whether prostate cancer becomes metastatic. Both Foxi3 and CXCR4 have been shown independently to make prostate cancer more aggressive, more invasive, and more likely to metastasize. While doing preliminary research, I found a study focusing on PC3 prostate cancer, on the prostate cancer cell line, PC3, in other words. PC3 cells are highly metastatic and express very high levels of CXCR4, much higher than DU145 cells, as you can see in this study. This makes PC3 an ideal model for studying metastasis-related pathways. The study also looked at CD44 and CD133, which are cancer stem cell markers that identify the small subpopulation of tumor cells that can self-renew, resist therapy, and drive metastasis, essentially. Cancer stem-like cells are associated with aggressive tumors, poor prognosis, and increased metastatic potential. So, our preliminary data showed that when Foxi3 is depleted, E-cadherin levels increased. E-cadherin is a hallmark in epithelial protein that is normally lost during EMT. So when Foxi3 is knocked down and E-cadherin goes up, this suggests a shift away from the metastatic phenotype. At the same time, we observed changes in CXCR4 expression, hinting that Foxi3 may regulate CXCR4. So here you can see a study that shows blocking CXCR4 with AMD 3100 reduces metastasis. This tells us that CXCR4 is not just associated with metastasis, it actively drives it. So blocking CXCR4 disrupts the cancer cell's ability to migrate towards CXCL12-rich tissues, like bone, in other words. Other studies suggest that when CXCR4 is depleted, osteoclast activity increases along with tumor growth. This highlights how complex the CXCR4 pathway is. It interacts with bone biology, immune cells, and tumor cells. So understanding how Foxi3 fits into this pathway could reveal new therapeutic targets. Other 4K proteins like Foxi3 and Foxi2 directly regulate CXCR4 expression. This supports our hypothesis that Foxi with an I, another 4K member that we discussed earlier, may also influence CXCR4. So Foxi1 and Foxi2 are like distant cousins of Foxi3. To test this hypothesis, we established a Foxi3 depleted PC3 cell line using lentiviral transduction. We isolated single cell clones, expanded them, and verified knockdown using real-time PCR and Western blot. Our hypothesis, or my hypothesis more so, was that downregulating Foxi3 would reduce CXCR4 expression and produce a less metastatic phenotype. Our objective is to establish a Foxi3 depleted cell line and examine how CXCR4 expression changes. So we began by culturing PC3 cells. We transduced them with the lentivirus that targeted Foxi3 or silenced Foxi3, the gene. Isolated single cells into 96 well plates, expanded them, and verified knockdown. This allowed us to generate stable Foxi3 depleted clones. We verified Foxi3 knockdown using RNA extraction, reverse transcription, real-time PCR, and Western blot. These methods confirm that Foxi3 expression is significantly reduced. Our preliminary data suggested that Foxi3 may directly influence CXCR4 expression. If Foxi3 regulates CXCR4, then targeting Foxi3 could be a new strategy to prevent prostate cancer from metastasizing to the bone. And as you can see in this graph, I put that they are directly correlated. Alternative approaches include using CRISPR, which I didn't include on this slide, for more precise gene editing or investigating other metastatic genes. And a key pitfall is that CXCR4 may be influenced by other pathways independent of Foxi3. So that could be pretty start over-ish. Like me just start over and try to find something else, which is also my alternative. Me just finding other metastatic genes. So it's important to interpret whatever results that I got correctly and carefully, and to also consider additional regulatory mechanisms. Like, as I read preliminary research, also just keep in mind like Foxi1, Foxi2, and then go down that pathway as well. Okay, so I try to go through this presentation as fast as possible so I could just pretty much go over some of the questions that were in class today. Kind of just touch on those a little bit more. I know I answered them all in class, but I think this is a good space to ask more questions and go over things a little bit more, essentially. Also, if you have more questions or comments, please email me and I'll be happy to talk with you about it in class on Thursday or after class on Thursday, either way. So one of the first questions was, does having high Foxi3 or CXCR4 mean that you have cancer? And I answered in class saying absolutely not, because Foxi3 is a gene that all people have, and CXCR4 is a chemokine receptor that also all people have. So it's only clinically important in the case of prostate cancer is because many cancers overexpress CXCR4 or Foxi3. Now, CXCR4 is directly linked to tumor aggressiveness and metastasis, meaning that CXCR4 is a guide or a driver for metastasis, essentially. So, high CXCR4 alone doesn't mean that you have cancer, or likewise with Foxi3. So another question was, why are CXCR4 and Foxi3 correlated, or why did I think that they were correlated? So number one, the first reason is EMT. EMT is a process, like I stated earlier, where epithelial cells become mesenchymal cells. So these tightly conjoined cells become more mobile and invasive. They become loosely spread around instead of tightly conjoined. And CXCR4 is strongly involved in cell migration and metastasis, right? So Foxi3, now entering the story, has also been implicated in regulating EMT-related genes. So if we know that CXCR4 is an EMT-related gene, and Foxi3 has been implicated in regulating EMT-related genes, that's how I kind of connected the dots. Also, the preliminary study I put earlier also provided some insight of Foxi3 and Foxi2 with the letter C that are distant cousins of Foxi with an I3. Why they are distant cousins, and that study pretty much gave me what I needed to draw a conclusion there, essentially. Well, not draw a conclusion, but form a hypothesis better. So yeah, after reading that study, it pretty much helped me to form a hypothesis about Foxi with the I3 and CXCR4. Another question was, how does Foxi3 work with an I? Foxi3 binds to promoter regions of DNA, and it activates or represses target genes. So the next question was, how does CXCR4 work? So CXCR4 is a chemokine receptor that directly correlates or directly influences cell movement, survival, and invasion. So the last question that I'm going to go over from class today was about E-cadherin levels. When we saw that E-cadherin levels were increased in Foxi3 knockdown cell lines, why did that happen? Okay, so E-cadherin plays a major role in epithelial conjoinment. It helps those bonds to be strong, and so that the cells are not loosely spread apart, right? So that by itself means that Foxi3 is a potential target, right? Primarily because it suggests a shift away from metastatic phenotype, meaning that this Foxi3 knockdown possibly could not, it's more epithelial-like than it is mesenchymal-like. Like we discussed earlier, mesenchymal cells are loosely spread, and so that gives them the ability to be more invasive, more motile, and high metastasis is linked to the depletion of E-cadherin, essentially. So with CXCR4 expression, when CXCR4 was depleted, E-cadherin levels were decreased. Let me make sure. Hold on. Yes, so after confirmation, this confirmed that CXCR4 being downregulated resulted in a E-cadherin level decrease. Now, this was a very great question, is because the decreased levels of E-cadherin associated with CXCR4 just means that it primarily may just influence, it actually really does influence cancer metastasis. So it just pretty much confirms all of the things that we've said before. And from this, I just drew a conclusion that I think that if Foxi3 was depleted, that CXCR4 levels would also be depleted, and that if Foxi3 levels were increased, CXCR4 levels would increase as well. So essentially, they play a role in helping one another out to make sure that the cancer metastasizes. And I believe that that pretty much was all of the questions that we had in class today. Like I said, I would be more than happy to clear up any more questions you guys have, or comments, or anything of that nature via email. And I hope you guys have a wonderful night, and I'll see you on Thursday. Thursday.